Okay, good afternoon, everybody. Thanks for joining to our second Tech Talk. Today, it's my pleasure to introduce two guests for the topic. The first one is Evangelina Lopez de Maturana. She had a degree in veterinary medicine from the University of Zaragoza and did a PhD in animal breeding and genetics at the Basque Institute for Agricultural Research and Development, also in Spain, and gathered some experience as postdoc in the Wageningen University of Research in the Netherlands, afterwards in the University of Wisconsin-Madison in the US, and finally also in the computational biology group in Melbourne, Australia. Since 2015, she works in the National Cancer Research Center, and among her topics of research is the integration of omics and non-omics data. Our second guest is Sergio Sabroso. He has a degree in mathematics, also from the University of Zaragoza, a master's degree in modeling in mathematical research, statistics, and computing from a collaboration between the University of Zaragoza and the University Basque country. He did afterwards a specialization in machine and deep learning techniques at the Instituto Tecnológico de Aragón and participated in some projects at the Institute of Health Research also in Aragón. Since 2020, he also works in the National Cancer Research Center with Evangelina, and among his topics is the application of artificial intelligence models, including medical imaging and different types of omics and non-omics data. Together, today they are going to present us the topic integration of multi-omics and non-omics data, artificial intelligence approaches and challenges. Please, we will have the presentation first, and at the end, there will be the round of questions. You can also write them if you want already in our chat, and afterwards I will read it aloud. Evangelina, Sergio, the floor is yours.
Thank you very much, Francesca, for your kind introduction. Good evening, everyone. We are Evangelina Lopez de Maturana and Sergio Sabroso, members of the Genetic and Molecular Epidemiology Group at the Spanish National Cancer Research Center. And we are grateful to the organizers for giving us the opportunity of talking about the integration of multi-omics and non-omics data, artificial intelligence approaches and challenges. During this seminar, we will cover the following aspects. First, we will give a brief overview of the state of the problem of integrating omics and non-omics data, and we will discuss the complexity that we will face at different levels. Then Sergio will briefly talk about the preprocessing of medical imaging data. And then we will present and discuss several analytical approaches for integrating omics and non-omics data. Then we will describe and discuss different challenges that we may face during this process, and then we will finalize this presentation with the present and future perspectives of this topic. During the last years, the technological advancements and associated reduction in costs have made possible to generate multiple omics datasets that help us to characterize the molecular processes of the biological systems. The integration of these omics data, in addition to radiomics, pathomics, as well as to other non-omics data, including clinicopathological, demographical, and epidemiological data, may help us to better characterize the exposome, the genome profile of the host, and also the phenome. An important step on this integration process is the data preprocessing. And during the last decade, several bioinformatic tools have been developed to preprocess omics data, and also multiple efforts have been made to improve the reproducibility of omics data preprocessing. As for the medical images preprocessing, Recent computerized approaches have been developed in order to extract extraclinical information, minimizing the human intervention. The two medical imaging fields that are catching more attention lately are radiomics and pathomics. And now Sergio, which is an expert in dealing with imaging data, will discuss on how to properly pre-process this data.
So thank you very much, Evangelina. As Evangelina has commented, in contrast to many omics data, images have a bit more of a special pipeline and must be worked with in a very different way. For this reason, and given now the great popularity that models capable of converting image into real numbers are gaining in the recent years, I will briefly explain how to work with image to use them in any subsequent model, and of course to integrate them with the rest of the data that Evangelina has mentioned previously. Normally, as Evangelina said, when we talk about images in the clinical field, we refer to two main types of images: radiological images, for example, CT, MRIs, whose extract features are called radiomics, or pathological images, where we mainly find the digital pathological slides, and whose extracted features are called pathomics. Both types of images more or less have a similar pipeline, except for some details in the preprocessing. But as we here in the lab are mainly working on radiomics at the moment, although in the near future we will move to pathomics also, we are going to focus on this field now. Thanks. This is the schema of any radiomics pipeline. As you can see, it consists of 4 phases. The first one is obtain the raw image without pre-processing. Once we have our images, the two most important points of the process start. First, the tumor labeling, also called the segmentation or annotation, where we select one or more region of interest from our entire image. And then the feature extraction, where we extract quantitative or qualitative variables, characteristics from this region of interest previously selected during the tumor labeling. As these are the two most important points of the process, I will explain them, exactly, Evangelina, in more detail. So yeah, finally, the last step, only to summarize, is for sure apply these features in any statistical analysis or model. Let's take a closer look first at what is this tumor labeling. As I mentioned, is selecting in a raw image one or more region of interest. Here, for example, you can see on the left that we are trying to differentiate or to segment in a CT scan pancreas and also pancreatic tumor. So, for example, here we are trying to delineate two objects of interest. This segmentation can be done manually, where you are delimiting on coloring, filling the different pixels of each CT with different software, or in more automatic way. I'm going to explain this automatic model in the next slide. But of course, it's worth to mention that tumor labeling is the most crucial and complicated part of the radiomics pipeline, because requires first a lot of care to perform the segmentation in the most correct way, and it requires a lot of time to do it. And in addition, it should always be performed by an expert radiologist, or at least supervised by one of them. So this segmentation, as I have mentioned, can also be done automatically thanks to certain deep learning algorithms, such this UNet. The UNet is a convolutional neural network, which is a deep learning model that works very well with images, with an encoder-decoder architecture here, which gets its name from the U shape of its architecture. I'm not going to go into the detail of how this algorithm works, but in very general terms, we give it a raw image as an input, and it returns the raw image and a mask with the segmentation as output. The main disadvantage here of this model is that all images need a lot of pre-processing to be included in the model. For example, think that if we are working with images from different centers, each of one will have a certain size, certain number of slices. If we want to work with every image in the same model, we have to perform a lot of pre-process. This problem was solved with the creation of the nnu-Net, thanks Evangelina, which provides an end-to-end automated pipeline. And to do that, use the same UNet architecture, but with this pre-process that I have mentioned, and also with hyperparameter tuning. I want to mention here that, like any neural network model, it requires first a training phase in order to use this algorithm automatically. And for this training phase, images that have been manually segmented will be needed. Once the model has been trained, when a new image arrives, the model is able to segment it automatically without the need of do it manually. Once we have the segmented image, we have to extract the features, these qualitative or quantitative values of the region that we have delimited of interest in order to be able to work with later in a model, or to be able to integrate them with variables from other fields. Again, here we have two alternatives. On the one hand, we have what is known as conventional and also traditional radiomics, whereby handcrafted features are extracted. These features try to describe the region of interest, capturing parameters that describe the morphology, the shape, the texture, for example, the volume, the maximum diameter, the maximum surface, the tumor compactness. For example, some of the most, the best techniques capable of doing this are the histogram equalization, the neighborhood Gradon difference matrix. And nowadays there is a Python package called Radiomics, which use these techniques. It's used to extract all these conventional features, and it's very, very easy to use. And for sure, on the other hand, we have what is known as deep radiomics, where again, convolutional neural networks extract the features in a more automatic way. And here some well-known architecture examples are, for example, the BGG, the ResNet. For sure, both techniques, conventional radiomics or deep radiomics, have their advantages and disadvantages. For example, with conventional radiomics, we always know what each of the features we are using really is. This is as opposite to the black box effect always present in deep learning models. But deep learning with radiomics, deep radiomics gives the advantages of discovering certain features, patterns, details, which are practically undetectable by the human eye. Although it should be noted that with the passage of the time, the advancement of the technology and also in computation, deep radiomic techniques are increasingly being used. Finally, with any of these two alternatives, we already have the qualitative and quantitative variables related to radiomics ready to be included in any known model. I'm going to show you very briefly an example of how the nnu-Net, this automatic segmentation technique, works in a project that we are developing here at the CNIO. In our case, we are training a model able to segment automatically pancreas and tumor from abdominal CT scans. In general, the results are quite good. Around 70% of the CTs have been correctly segmented in automatically way, for sure. And some radiologists did not find any detail to modify. And as I have mentioned, it happens with the vast majority of the CTs. But unfortunately, thanks Evangelina, and as with any model of artificial intelligence, there are some CT scans that have not been segmented correctly. For example, this patient, the algorithm segmented a tail tumor, and exactly, if you go exactly, and yeah, when in our lab together, the model segmented a tail tumor, but when our lab together with the radiologist, we detected that it was a head tumor. So here the model is totally wrong. But in general, also we have to be careful. It's a tool that is working very well, and the people who are segmenting the CTs in our lab told me that thanks to this only had to need to review the segmentation and not start from scratch, saving them a lot of time and efforts. So we have already seen how we can work with images and extract their characteristics. And now Evangelina will tell you about several techniques capable of integrating these variables with the rest of the fields of interest.
So once we have processed the omics, radiomics, pathomics, and non-omics data, now we need to integrate them in a statistical model. And to integrate this high dimensional and heterogeneous data requires the application of more advanced statistical methods such as the AI algorithms. So the application of these algorithms implies several challenges, not only at the methodological level, but also at the biological interpretation of the results. And also, when we integrate this plethora of omics, radiomics, pathomics, non-omics data, We are going to face also challenges related to the nature of this data. They have different nature, and also we will face to the fact that there is a relationship, or there may be a relationship between these data types. So in the upcoming slides, we will provide some examples of the application of AI algorithms to integrate high-dimensional multimodal data. In the last year, several initiatives have been proposed to overcome the p-value dictatorship in the genome-wide association studies and to incorporate functional information to interpret the role of the genetic variants associated with a given disease. In a paper published last year, we complemented a new pancreatic cancer GWAS, here the 1D approach, with an alternative prioritization of variants using a spatial autocorrelation method that we call the 2D approach. So by capturing the LD structure of nearby SNPs, this 2D approach leverages the values of SNPs with low minor allele frequency. Importantly, we also integrated the high-CMAP interactions. We called this phase the 3D approach to gain additional insight into the inherited basis of pancreatic cancer, overcoming the challenge of ascribing variants in non-coding regions of the genome to the corresponding regulated genes. Additionally, we also performed a functional in silico analysis at different levels, at the variant, gene, and pathway levels, using publicly available information to further prioritize potentially relevant candidate variants for pancreatic cancer risk. So this approach helped us to identify 17 new pancreatic cancer susceptibility loci. Some of them were relevant in pancreas physiology or carcinogenesis, such as NR5A2 and XBP1. And others didn't have any known pancreas-related function, as MN1. Note that these last two susceptibility regions were identified only through the 3D approach. We believe that similar integrative approaches could be extended to the prognosis prediction field using other omics in the tumors, which is also of interest for our group. In a recent review published in Nature Review Papers, Wang et al. Showed other alternative approaches integrating functional information through machine learning algorithms. An example is the NetWAS, and in this method, so this method takes as input the marginally significant GWAS hits as positives and the lower ranked genes as negatives, and uses the tissue-specific network H genes, weights, as the feature set in a support vector machine classifier in order to reprioritize the candidate genes. Networks can also be used to analyze the connectivity patterns of known disease genes in order to obtain accurate and relevant predictions, because the dysregulated genes underlying disease are frequently involved in tissue-specific processes. Moreover, these genes can be partitioned into modules that can be further characterized based on geo-enrichment analysis. We have also deeply explored the application of other machine learning algorithms that also allow us to integrate highly dimensional data, such as the Bayesian Kernel-based regression methods. So very briefly, these models are able to reduce the dimensionality of a problem. So imagine that we may have hundreds of thousands of SNPs, and this number is much greater than the number of individuals that we may have. So with these methods, we are able to reduce the dimensionality of our problem to an n by n problem, where n is the number of individuals. Here you can see the application of this method to deal with hundreds of thousands of germline genetic variants. And note that the application of this machine learning algorithm involves the computation of a genomic relationship matrix, where the off diagonals indicate the pairwise genomic similarity between two different individuals, I and J. As a result of this model, we will obtain an estimate of the genetic risk score of the individuals, and also we will have the estimates of the genomic and the residual variances. We may extend these models to the analysis of highly dimensional multimodal data, as in this example, where we integrate high dimensional genotype, also radiomics features that were extracted, as Sergio commented before, and also expression data. For this particular example, we would need to compute 3 different kernels, one for each data type, and the computation of these kernels or these similarity matrices would differ depending on the nature of the data. Oops, sorry. Here you can see the performance of different multi-kernel-based regression models that were applied to pancreatic cancer risk prediction, in which we integrated high-dimensional multimodal data. And these are results that have been obtained in our lab. So we applied this strategy to integrate microbiome data obtained from different locations, from oral and fecal samples, and we evaluated the predictability of each microbiome risk score. As you can see here, the best prediction was obtained with the risk score based on fecal microbiome data. And in terms of prediction, adding oral microbiome data did not improve the classification performance of fecal microbiome data. We also integrated genotyping and DNA methylation data of the HLA region with oral microbiome data and estimated the percentage of the pancreatic cancer risk that was explained by each of these components. As you can see here, the DNA methylation data is the one that explains a larger proportion of the phenotypic variance. And moreover, we also integrated the DNA methylation data, and we incorporated the interaction with diabetes and smoking status that are the most important risk factors for pancreatic cancer. As you can see here, we observed that the interaction term between DNA methylation and diabetes status explained 33% of the phenotypic variability associated to pancreatic cancer risk. Machine learning methods have also been applied to model transcriptional effects of non-coding variants and can help also to predict their impact. And here you can see a list of these bioinformatic tools that in the majority of the cases, they use deep learning methods. One of them is the DeepC framework, which is a convolutional neural network-based approach where the deep learning model was trained using genome-wide chromatin features that impact transcription in more than 200 cell types. And this deep learning model can help to predict the consequence of a nucleotide substitution in the genomic sequence. Now, Sergio will introduce you other state-of-the-art machine learning methods which have not been widely applied in the biomedical field, such as the Ensemble methods. And this methodology is currently on the rise and can be perfectly used to integrate different types of data.
you very much again, Evangelina. So I'm going to present this emerging methodology we are exploring now called, as Evangelina said, Ensemble learning. These Ensemble methods combine predictions from different models, both simple models and machine learning ones, with the aim of improving the predictive capacity of an overall model. This methodology is based on the idea that the disadvantages of a single model can be overcome with the strength of the others. This family of methods, we can classify it into three. Bagging, where we apply the same model, but for different subpopulation and then combine them. This model is this kind of methodology is very useful when we have a small sample size. Stacking, where we use different methods on the whole population and then we combine them. And boosting, which assigns different weights to the population to give them different degrees of importance depending on the interaction, assigning a higher weight in the next iteration to those individuals that have been predicted to be worse. These models, as I have mentioned, can be of any type, including for sure neural networks. Currently, there is a model that is achieving very good results in various fields, but which for the moment has rarely been applied in the biological field. This method is the AdaBoost CNN, which is based on the AdaBoost, one of the most successful boosting methods. And here we initialize the weights uniformly from the entire population, and we create a first convolutional neural network. We train this network and make the first predictions. Based on these predictions, we update the population weights for the second iteration and create another convolutional neural network. In addition, the starting weights of this second neural network are the ones we get pre-trained from the previous one. And this is repeated an arbitrary number of times until we have the final model. This methodology is also starting to be used in feature selection, although it should be noted that there is a few related literature so far, at least in the biological field. In these cases, ensemble methods can be applied in two different ways. On the left, we can use different feature selection models on our original dataset and then combine them all to get the final model. Or, for example, on the right, we can be a bit more novel and combine this, for example, with a genetic algorithm. Here, the genetic algorithm selects a subset of features to be evaluated with different methods: a logistic regression, a super vector machine, a neural network, naive Bayes, and a decision tree. All the predictions here are combined and merged. And this combination will give me an evaluation of the subset of features that it's sent back to the genetic algorithm, which will give us another subset of features to be evaluated. This is again repeated until the algorithm converges or until a fixed number of interactions, and the genetic algorithm will give us the optimal subset of features. This ensemble method is another methodology that we can apply, but it's also important to highlight that we are dealing with a very complex problem. It's the integration of multiple data, which gives rise to a series of challenges that we have to take also into account. For example, here, the correlation and the interaction between data types, the disease heterogeneity, the population substructures, the fairness between the different sets of data. For example, the statistical power, we have much more features than individuals, the shortage of statistical tests, the lack of studies to validate replicate results. One very important is the dynamic nature of the health and the pathological process, because in all these methodologies, we are not taking into account the time dimension that for sure considerably affects the development of these processes. And of course, and this is common to many problems of this kind, the economic cost involved, which is not always easy to achieve. We are applying some of these techniques to integrate all this data in PanCAME. PanCAME is a European consortium in which our group is collaborating with the aim to improve pancreatic cancer treatment with artificial intelligence, optimizing and integrating genomics and medical imaging. For more information about the consortium, please visit its website. So for sure, that PanCAME will give us a unique opportunity to apply these artificial intelligence models and integrate multimodal data. Note that also the methods that Evangelina and I have mentioned are on rise and have a high predictive capacity. We are still far from the personalized medicine that we want to achieve. So in the future, we think to combine maybe these strategies with new large-scale techniques, including, for example, real-size simulation, such as the biomodels, the digital twins, or even gigascale simulations. And that's all. Thank you very much to all of you for your attention. This is a small part of the things that we are doing now at the CNIO, specifically in the genetic and molecular epidemiology group, of which Evangelina and I are members. And we would like to thank them, and in particular to Nuria Malaz, who leads the group. If you have now any questions or comments, we will be very happy to answer them. Thank you very much again.
Thank you very much, Evangelina and Sergio, for the nice presentation. Please, to all the people that are present, feel free to start asking questions, either writing them in the chat and I can read them aloud, or maybe we can try raising hands and you can also say it aloud. Just maybe introduce yourself first so Evangelina and Sergio also know a bit who you are. Okay, maybe I start with the first question. Sergio, I'm a little bit curious. When you mentioned the process and all what is required for the imaging, to go from imaging information to this number information, you need to train the model.
Yep.
How long more or less take this training?
Depending of the computational power that you have, and depending also the number of images that you are including in the model. But for example, one model about 100 CTs, which more or less computational normally, not too high machines. We are talking about more or less 1 day. 1 day per fold. Usually we train more than one fold, so like five days more or less. It will be a complex model. Yeah.
Okay, that's not so much time. Then you can use it and apply it.
Yeah, because we are talking about, for example, in our case now we are working with 100 CTs, and it's not very easy to collect CTs. But if you have, I don't know, if we are talking about big data and you are working with a lot of images, for sure the model will be very expensive regarding time. Yeah.
Thank you. Okay, perhaps I can ask a second question. I don't know if it's better for Sergio or for Evangelina or for both. When you are combining all the variables, so from the omics and non-omics, do you have already an idea which is the best way of, so which will the order or hierarchy to include this information in all this formula that you need for combining all the results or the variables at the end? Is there any idea already, or is just trying and failing, like we say?
Yes, as for the Bayesian Kernel-based regressions, so you may include all the data of the different nature that you may have, and you are analyzing them simultaneously. So for example, you may perform an initial approach and see if, for example, there is a data type that doesn't explain much of the phenotypic variance. So you may drop that from the model, so it's more parsimonious. And regarding the deep learning, maybe Sergio can.
Yeah, in general, we include all the omics data and non-omics also at the same time. Maybe you can give different importance if you, for example, in a previous analysis, you saw that one feature is more related with your final outcome, maybe you can give different weights, but normally we include everything at the same level and at the same time. That's the idea.
Okay.
Also, you can perform feature selection if you want,
Sure.
in order to reduce the dimensionality of the problem and select the features that are associated with the phenotype that you are interested in, discarding those that maybe are just adding noise. So this is something that you can also do in order to have a more parsimonious model and to work with a lower number of features.
Okay.
For sure, in this kind of models, you need this previous feature selection, but normally the idea is, again, in the feature selection, work with the same conditions per each type of data at the beginning.
Okay. Yeah. Thank you. We have a question from the people. Sorry, the first one first. When you integrate different perspective, you perhaps had a lot of features that turned up. Was this a problem, an issue, and how did you deal with this?
We already commented that the feature selection process would be nice to apply before integrating them in a model. So also you may perform a cross validation. So you may perform this or you should do it in a cross validation scenario in order to avoid overfitting. And then you may perform different strategies on the feature selection to select those that perform better classification in the cross validation scenario. This could be a strategy.
Thank you. Regarding to the data storage, is the data store in one place or in one database for different omics levels? And if so, how do you deal with metadata across the omics levels?
So we usually have different databases for each data type, and then we combine them all together in the same place once we perform the feature selection or once we want to integrate them in the same model. I don't know if I answered the question.
Yeah, I think it's the answer. We have another question regarding to the balance of the performance of the machine learning in terms of accuracy. How do you deal with this? For example, how do you balance between minimizing false positive or negative, or true positive or negative? How do you deal with this issue?
Yes, that's a very good question. For example, when you want to identify or to prioritize genetic variants that may be associated with a given disease or condition, so it's true that you need to find a balance between the false negatives and the false positive. What we did, for example, in the paper that I commented that was published in Genome Medicine, what we did is to add several layers of information in order to give a list of prioritized variants that had a functional relevance apart from the statistical p-value, right? So this is if you want just to make inference, but if you want to make predictions, so then you may end up with the best combination of features that will give you the best classification performance.
Okay. We have a question regarding to considering model prediction uncertainty, either in the initial semantic segmentation model for the CT scans or in the Ensemble models.
Yeah, for sure at the beginning, if you want to train, if we are talking, for example, regarding one image model at the beginning, your predictions will be very bad because, I mean, you can use, for example, pre-training models, but the good idea or the best idea that you can do here is, instead of you are using these pre-trained models, you can also train again your weights and update your model. But yes, at the beginning, for sure, that your model, the prediction, it will be very bad, but you are training the model. At the end, your final model will be more or less well. But for sure, it's regarding, for example, the imaging, it's will be necessary to be supervised by a radiologist and only to be a helpful tool to see the segmentation correctly or not. That's more or less the idea.
Okay, we have another question. 1 second. No, you just send me the link.
By a radiologist and only to be a helpful tool to see the segmentation correctly or not.
Sergio, so we have another question in the chat about model interpretability in the case of the Ensemble modeling methods. It makes sense to aggregate predictions and make superlearners. Does it make sense to aggregate measures of importance from methods that support this?
This is a very good question. Yes, here to evaluate, for example, these example methods, we are learning on this part. So here I'm not totally an expert for sure. But yes, for example, you can aggregate all these measures. You can combine, for example, when we combine some methodologies, the idea is, for example, perform a mean accuracy or a mean AUCs from the several models. And yes, it's not easy how to aggregate these measures, because if you are using some alternatives, you are considering different things. But yes, this is a very good question. But the idea is, yes, at least at the beginning, integrate and for example, aggregate and perform the mean of every methods. And maybe the most important thing is, maybe if you are evaluate each method independently, you can, maybe if you see that it's one method is stronger in one part, maybe you can give them more weight in these predictions or in these classifications. So yes, it's not easy how to consider each final accuracy measure for each model. We are learning about that also. It's not easy. Thanks, John.
We have a question, a bit more clinical, perhaps. If it's that you are taking into account neoadjuvant therapy in your research.
I think we are considering both, Evangelina, neo and also adjuvant therapy in the patients that we are working, for example, with CTs. In my case, yes, there are some patients treated with neoadjuvant and there are others treated with adjuvant. So for sure, then it's a good also alternative when you are considering this. For example, only if I'm talking about radiomics, it's also interesting that at the end you can combine also these clinical features, trying to adjust your final model and consider also if you are if your patient has different treatment, has different clinical features, and this is a very good point of the machine learning methods, in particular here the deep learning ones. Yeah.
And also related to this, another challenge that I don't know if we commented is that sometimes we have like a Gruyere cheese because we don't have all the data collected for all the patients, and sometimes if we want to combine different studies, we don't have the information for all of them. Also, when we combine different or patients recruited in different hospitals that may have treated in different ways, So this is also a challenge that we may face. So sometimes it's not that easy, for example, to include the treatment, or sometimes combining different hospitals, they don't have the same treatment regime. So so this is something that that we need to consider also when we apply these models.
It's very difficult to integrate all this type of data, but it's more difficult if it's possible integrate data from different repositories, from different studies. This is also a very huge challenge. Yeah.
Yeah, thank you. Do we have any other questions? Okay, if not, I think everybody has enjoyed the presentation. I got also all the messages here of thanking for the nice presentation and the very interesting topic. And yeah, we hope you can continue with this modeling. It looks very promising and for sure very useful, because it's, as you just mentioned, very difficult to combine all this information, omic and non-omics, and on top of that, the challenges that Sergio just mentioned also at the end. So yeah, thank you very much. And yeah, we hope we can hear from you in another opportunity.
Well, thank you very much for giving us the opportunity, and thank you for the nice questions.
Yeah.
Okay.
Thank you very much, all of you.
Bye-bye.
Thank you. Bye-bye. See you later.
Bye.